What this project hopes to achieve is to understand why mammographic density is a risk factor for developing breast cancer. And to do that, we're using animal-free models using human tissues. There's clear advantages because if we're studying a human condition, we need to use human tissue, and that's exactly what we're doing here in Aberdeen. Animal tissue is clearly very different from human tissue, and particularly the project that we're looking at the fibroblast cells. So a fibroblast is a type of cell which is found within breast tissue and indeed other types of tissues within the body. It's a supporting cell, if you like.
We take fibroblasts and generate those cells from the tissues that have been donated to us, and then we use them in place of taking animals, the cells from animals, and use these models instead of implanting human cells into the animals. My research is looking at these environmental chemicals known as endocrine disrupting agents. One of them is bisphenol A, which is shortened as BPA. And this is found in plastic water bottles. Other ones include ones that are found in the diet. One of them is genistein, and the other one's resveratrol. And genistein is found in soy products, and resveratrol is found in things like grape skins and sort of red wine. And we're looking at how these affect cells in the breast known as fibroblasts.
It's very difficult to do these types of experiments in animals because if you implant human fibroblasts into animals, they very quickly get placed by mouse fibroblasts, so that renders a little bit of a question mark over how valid the model is to study a human disease. But if we can use human tissue instead of animal tissues, then we're likely to be much more confident in the results that we get. Typically, three-dimensional cultures have been done using collagen. The source of collagen actually is usually from rat tails. We have to look at other alternatives to using collagen, and that's why we've been lucky to have come up with a culture method which allows us to grow the cells in 3D, and it's called Perfusion Pal. Perfusion Pal is a system that allows us to grow our cells in 3 dimensions as they would in the body. It has a continual flow of what's called an artificial blood substitute that flows over the cells, which is exactly how cells would find themselves within the body. So that's a really new model that we're starting to develop. The second model that we're using takes cells from human breast tissue, and we can combine these cells in the laboratory such that we can recapitulate what human breast tissue looks like. And we always use that as our gold standard. So we have our human breast tissue, we know what it looks like, and we hope to be able to have our models looking very similar. Then what we can do is to change the genes that are produced by one or other of the cell types and see what effect that is going to have on the behaviour of other cell types within the model. And ultimately, we want to be able to use our endocrine disrupting agents within that model as well to see how that can affect the behaviour of the different cell types within the model.